everyone. First off, I want to apologize that our lunch order wasn't placed. I sent it in on Friday, and I think there was some confusion about what day it was being held, so I'm sorry. We're not able to offer the lunch half of the Lunch and Learn seminar today. We did have a lot of registrants for this seminar too, so I'm very sorry again that we weren't able to give lunch to you all. Today's seminar is entitled Gone Metabolic, Fishing for Secrets with Seahorse, or something along those lines. We're going to hear from Wojtek of the cytometry shared resource, and then two Wilmot investigators who will go into how they've utilized the Seahorse analyzer for some of their research projects. And my name's Tracy McGinnity. I work in shared resources management. So if you have any questions about any of the shared resources, I'm happy to answer them. And finally, this seminar series was supported by Vivian Palladoro and her endowment fund. She's usually the woman that joins through Zoom. Sometimes she has her magnifying glass. She's not able to attend today, but she is the one that helps us put on these seminar series for everyone. So with that, I'll hand it over to Wojtek to give a brief summary.
Hello, hey, well, thanks for organizing it, even though we're fasting today. I guess it's for the good. Okay, so we got this idea, but perhaps Seahorse that we host and our facility needs a little bit broader introduction, and so we decided to organize this seminar and also invite excellent speakers for today. So I'm Wojtek Wojciechowski. I work at Flow Cytometry resource, but I'm kind of in charge of the Seahorse and some other non-typical instruments there. So maybe we'll just start with the talk. So today we have Paul Brooks, who will be talking first. And I actually know Paul for some time because he was on the committee for our HTS core, that kind of, it worked, but not for too long. But Paul actually is originally from UK. He did his PhD in biochemistry at the University of Cambridge. And then in '98, he moved to US and was doing his postdoc at the University of Alabama at Birmingham. Right? Since 2003 in Rochester, assistant professor of anesthesiology. He's studying metabolism, of course, and mitochondria in the setting of heart attacks, so very important subject. Published over 160 papers, and interesting fact that I just learned, he is also interested in forensic detection of and reporting of image manipulation and research misconduct. And that actually led to some threats of lawsuits, but none of them succeeded, so I think he's doing a very good job. And the main reason he's here is because he's a true expert in Seahorse. He had the first ever Seahorse machine ever built here at U of R in 2009. So please show us what it is.
Thanks. Okay, thanks, Wojciech. So we're going to go back to undergraduate biochemistry a little bit today. So this is metabolism. This is sort of a recap on everything that happens in the cell when we're talking about metabolism. So basically at the center of the universe here is the mitochondria. We have glucose breakdown through glycolysis, GAPDH being a critical step that requires NAD. This makes pyruvate, and then we can do two things with pyruvate. When we have oxygen, we can send it into the mitochondria and then into the Krebs cycle. If we don't have oxygen, we can't do that, so we turn it into lactate, and then NADH gets recycled back to NAD to allow glycolysis to continue. We can also send glucose down the pentose phosphate pathway. We don't really have an ability to measure this using the Seahorse. The reason for doing this, of course, is to make ATP. If we do glycolysis, we get 2 ATPs. However, if we send the pyruvate into the mitochondria and all the way through the Krebs cycle and oxidative phosphorylation, we get 36 ATPs. So this is a much more efficient process. And of course, the other thing we have is something called the malate aspartate shuttle for sending this NADH into the mitochondria and back out again. And in addition to pyruvate and the product of glycolysis, we have a lot of other fuels that we can burn: fat, ketone bodies, branched-chain amino acids. These all feed into the Krebs cycle to make reducing equivalents like NADH. These pass electrons onto the respiratory chain, aka the electron transport chain, aka oxidative phosphorylation. And this is where all the oxygen gets consumed in your body. There are other things we can do, such as bring carbon out of the Krebs cycle to make citrate, which is used to make fat and other types of things. So in fact, in cancer cells, for example, they bring glutamine in, drive metabolism through the Krebs cycle backwards to make citrate in order to build lipids to make membranes. So this is all sort of textbook biochemistry, and it all builds upon this sort of seminal discovery by Peter Mitchell, who won the Nobel Prize for this a long time after his paper reporting it. And the key discovery of Mitchell is that the intermediate that links the oxidation part to the phosphorylation part is the proton gradient. So we have a respiratory chain here, which is using electrons. It's passing those electrons onto oxygen to make water. The electron transport chain is a series of electron-driven proton pumps. They pump protons out to make a proton gradient or a membrane potential. So your cell membrane potential is anywhere between, say, 40 and 70 millivolts. In the mitochondria, the membrane potential is about 180 millivolts. That proton gradient is then used by ATP synthase to turn ADP into ATP. We can measure that membrane potential in millivolts. Like I said, it's about 0.2 volts across the membrane, so much bigger than the plasma membrane potential. And the reason this caught on as a model is it explained, unlike previous models of how mitochondria work, it explained the action of a series of chemicals which were called uncouplers. So dinitrophenol, FCCP, these are chemicals that carry protons across biological membranes. And so a lot of the previous models for how this worked before Mitchell came along didn't really have incorporation of why dissipating the proton gradient across the membrane would uncouple the oxidative bit from the phosphorylation bit. And so these uncouplers, this is what they do. They dissipate that proton gradient so that you can burn fuel without making ATP. Not surprisingly, there's a lot of interest in these molecules as dietary drugs to combat obesity and this kind of thing. None of them have really worked very well. Mitchell is an interesting guy. He was independently wealthy, owned a big manor house down in Cornwall in the tip of England. He had his wife was his lab technician. He ran a lab in his basement. He wasn't affiliated with any university or anything, so he was sort of rejected by the scientific establishment, and just ran his own experiments at home, and had a bit of trouble getting them published into journals. But he was right in the end. So the other tool that was essential for this, to discover this, was this tool called the Clark oxygen electrode. That was invented by Leland Clark, who's from Rochester. And the person who did a lot of this work was Britton Chance, who was also an Olympic gold medal sailor. And so the type of experiment would be you'd place mitochondria in a small chamber, and you measure the concentration of oxygen over time. So as mitochondria consume oxygen, the concentration of oxygen goes down. So here's our hypothetical mitochondria, and here's a buffer and some mitochondria. We put them in the chamber and nothing happens. Why? Because we haven't given them any fuel. We haven't given the mitochondria anything to burn. So when we add a fuel, something like succinate, to feed into the Krebs cycle, to pass electrons onto the respiratory chain, now we start consuming oxygen. And so Chance referred to this as state 2. Why are we consuming oxygen? So we're consuming oxygen here and pumping out protons. However, we're not consuming oxygen very quickly, because what happens is the proton gradient builds up, and the proton gradient ends up feedback inhibiting this process. So we can't consume oxygen very quickly. You'll notice that what happens when we add ADP. So ADP is the substrate for the ATP synthase to make ATP. And when that happens, we basically open the gate for protons in the ATP synthase. That dissipates that proton gradient, and that then allows those pumps to go much faster. Okay? And so this is known as state 3 respiration or phosphorylating respiration. When we run out of ADP, we go into a state which is state 4. For technical purposes, state 4 and state 2 are basically the same. And in addition to, for example, opening the proton gate by giving ADP, by the way, in the cell, the way you would give ADP is to do some work, right? Turn some ATP into ADP. In addition to that, we could also just add an uncoupler and get a maximum rate of uncoupled rate. And so this was the classical way that mitochondria was studied in the 1960s, and that led to this sort of model using various different pharmacologic inhibitors of respiration, this model of the respiratory chain, where we have the Krebs cycle, passing electrons to complex 1 and complex 2. Those complexes pump protons and send electrons to the coenzyme Q, or ubiquinone. This passes electrons to complex 3 and cytochrome C, and then complex 4, and then oxygen. So technically, complex 1, 2, 3, 4 are called the respiratory chain, or respiratory complexes. Complex 5 is the ATP synthase that's going to use that proton gradient to make ATP. It's not a respiratory complex, but it is a complex. And so this model, up until probably about a decade ago, this is what most people thought of when they thought of oxidative phosphorylation. So basically what came along in 2009, and this is the plate, you can pass this along and play with it. If you haven't seen what the plate looks like, that's basically what they are. This is the 24-well version of the machine. Like I said, we had the third instrument that was built in 2009. Basically what this is, it's a high throughput version of the oxygen electrode. So it consists of two parts. You have a well, and the cells that you want to study are on the bottom of the well. The key thing is the cells have to be plated and stuck to the bottom of the well. They have to be adherent. And then the green thing that sits on top, this is a disposable cartridge. And on the tip of these little probes here are sensors that are basically porphyrin-like molecules that are responsive to oxygen and pH. So these are sensors, they're fluorescent. In fact, the oxygen sensor, its fluorescence is quenched by oxygen, so it's negative response to oxygen concentration. And you'll notice the well here is not perfectly straight-sided. The well is tapered in size. So this is some cardiomyocytes stuck to the bottom of a well in a Seahorse plate. And you can probably see it easier on the plastic demonstration you have. But within the top of the plate, there are 5 holes per well. The middle hole here is where a stainless steel rod goes down, and this contains fiber optics, which interrogate these fluorescent probes. And then around the outside, there are 4 little holes, which are injection ports, which get loaded with drugs, which can be added to the chamber. And there are essentially 2 positions for this plate. The whole green thing moves up and down relative to the plate of cells. So in the upper position, the cells are exposed to the whole of the media in the plate, 100 microliters, 150 microliters in this case. And they're just sitting there in media doing nothing. Now, when the machine basically says read, the plungers come down, and what that does is it seals the cells into what is called a transient microchamber. So you're basically taking these cells that were previously sitting in 100 microliters of media, and you're squashing them down into 3 microliters. So in the 96-well version, the volume here of this little headspace above the cells is about 3 microliters. If you look ever so carefully, you'll see on the bottom of the well, there's actually 3 little dimples sticking up from the bottom of the well. Those are actually there to stop the probe from squashing the cells. That's to hold the probe up off the bottom of the well. And the height of those dimples determines the volume of that chamber. So what you're doing basically is trapping the cells in a finite volume of oxygen, of buffer. And the cells are going to consume oxygen, and when they do that, the oxygen concentration in that space is going to go down. And that enables you to measure oxygen consumption. And then when you're finished with the measurement, you lift the plungers up, the cells get flooded again with this big bulk of media, and then, for example, you can inject a drug from one of the ports. After you've injected the drug, in fact, the plungers will move up and down to mix the drug into the media, and then you can go on and take another measurement. So the key things about this, it uses cells, it doesn't use isolated mitochondria. The cells have to be adherent to the bottom of the plate. And there's a workflow that we're going to describe in a while. So this is what the raw data looked like coming off the plate. Typically, you get about 5 data points a minute, 12 seconds to cycle through the whole plate. This is oxygen over time, and you need about 2 minutes in order to get a slope on this line to measure oxygen consumption. In this case, this is a couple of different measurements, followed by injection of a drug in port D, followed by another measurement. You can inject drugs and you can mix in between reads. Typically, you need about 3 readings per segment. We'll talk about why that is in a sec. So this is probably the Seahorse diagram that most people are familiar with. This is something called a mitochondrial stress test assay, also known as a BOFA assay, or that stands for the different drugs that were used. And so the BOFA assay, basically what we have now is, instead of our oxygen consumption, where we have oxygen over time, the machine has essentially already calculated the slope of this line. So the critical thing in the BOFA, or the mitochondrial Seahorse results, is the y-axis is a rate, OCAR, the R of OCAR, oxygen consumption rate. So you have a rate. That rate has been calculated automatically from this type of trace. So that we're not looking at the slope of this line. Ideally, these 3 readings are all under the same condition, and there shouldn't be a slope here. They should be the same, because we're just measuring the same thing 3 times to make sure that the cells are stable. So this is called our basal respiration rate. We then add oligomycin. So oligomycin is a blocker of the ATP synthase. So we block the ATP synthase. That's equivalent to putting the mitochondria in the cell into that state 4, or sort of quiescent state. So you see that when we add oligomycin, you get a decrease in the rate, and that decrease in the rate basically tells you how much of the oxygen consumption at baseline was being used to make ATP. Right? There is some discussion as to whether the residual oxygen consumption that's left over is the proton leak. That's a very complex argument. We're not going to get into that today. So what we've done is basically here, we've got cells, they're respiring, they're consuming oxygen, and we've asked a simple question: how much of that oxygen is actually being used to make ATP? Now, cells, under normal conditions, don't use 100% of their capacity. So, for example, if you're driving along the road at 20 miles an hour, you're using maybe 20% of the capacity of your car. Your car could go 100 miles an hour. You're not using the full capacity of the engine. In the same way, everybody has a heart rate anywhere between 40 and 70 beats per minute, but your heart rate can go up to 220 beats per minute, right? You have what is called a reserve capacity. And so the next step in this BOFA assay is basically to add an uncoupler. So when you add an uncoupler, going back to our sort of state 3, state 4 model, when there is no ATP synthase working, the rate of oxygen consumption is essentially, it can only go as fast as ADP is being made available. So how do we discover what the maximum respiration rate would be? So the way we do that is to basically add an uncoupler. So when we add an uncoupler, that's going to allow the respiratory train to run at maximum capacity. It's essentially like sitting in your driveway in your car in park and revving the engine. We're not going anywhere. We're not using that energy to do any work. We're just asking how fast can the engine go and looking at the rev meter. or the tachometer on the car, okay? Apologies for people who don't drive or don't know what I'm talking about with cars. And so what you see that when we add the uncoupler, we get a maximum respiration rate. This is essentially the maximum capacity of the respiratory chain. We then can do something interesting and look at what was the baseline respiration and what's the maximum respiration, and that's going to tell you what is the spare capacity. And it turns out that's quite a useful parameter that's highly variable between cell types. So for example, in the cells that my lab studies, cardiomyocytes, there's about a 3 or a 4-fold excess capacity. So at baseline, those cells are only using 20 or 30% of their respiratory capacity. They have a big headroom, right? In other cell types, actually, so if you look at endothelial cells, for example, they're using about 90% of their capacity at baseline. They have no wiggle room. They have no room for extra work, right? That means those cells are going to be more responsive to stress, less able to deal with a change in energy demand, right? And so that spare capacity, it turns out, is quite an important parameter for pathology and thinking about how cells respond to stress. Because the closer you are to that maximum, the less headroom you have, the less wiggle room you have in order to meet an energy demand that comes along. And it turns out, actually, that a lot of pathological insults, drugs, oxidative stress, things that cause cell death, damage to mitochondria, what they do is they damage that spare capacity. So what you're doing when you insult mitochondria is you're getting rid of the spare capacity, and so the cell is now operating closer to its maximum because the maximum has been reduced. And that's a fairly common thing that's seen. Now, at the end of the experiment, we then add a couple of inhibitors that completely block the mitochondrial function. That collapses the oxygen consumption down. Now, there are processes within cells that consume oxygen that are not mitochondria, right? Anybody that studies macrophages, inflammatory cells, you're going to have, respiratory burst, NADPH oxidase, these types of things. So this non-mitochondrial respiration is usually corrected for at the end by subtracting it away. There is something else that we need to mention, which is that not only is this an oxygen sensor, there is a pH sensor. And so glycolysis, the process of glycolysis, makes acid and protons. And so there is another parameter that the instrument will give you, which is ECAR, which stands for extracellular acidification rate. Basically, how fast is this cell producing acid? Now, it turns out this is a little bit more controversial because glycolysis is not the only thing that can make acid. In fact, the Krebs cycle makes carbon dioxide, bicarbonate, carbonic anhydrase reaction. So there is actually quite a big contribution to acidosis from the Krebs cycle, not just from glycolysis. So the Seahorse people sell this as sort of a glycolysis assay. It's not really a glycolysis assay. But it's based on the same kind of principle. You can give glucose as a substrate. You're going to get a certain rate of acid production. If you then block the mitochondria, the cell now cannot use mitochondria to make ATP, so it becomes more reliant on glycolysis. And so it's going to ramp up the rate of glycolysis, and this gives you a glycolytic capacity, and then gives you a glycolytic reserve. And then you can do something like put in 2-deoxyglucose at the end to block glycolysis. The discovery that sort of happened probably 5 or 6 years ago now is in fact, right at the beginning, we talked about this idea that glycolysis makes 2 ATP and mitochondria makes 36 ATP. So the assumption was, like, how in the hell can glycolysis ever make up for mitochondrial inhibition? And the thing that's really becoming apparent as people do more of these types of experiments, the dynamic range of glycolysis is absolutely massive. The ability of glycolysis to ramp up, glycolysis would have to increase 16-fold in rate to make up for the ATP that is lost when the mitochondria is inhibited. That's not a problem. Glycolysis is perfectly capable of doing that. Glycolysis has a huge dynamic range. And that's something that's really only been appreciated in the last 5 or 6 years. The other thing, yeah, so this also talks about this. We have CO2 coming from the Krebs cycle as well. So the workflow for this is actually fairly complex. There's one of these plates right here. So essentially, there are a few things that I want to talk about that are important if you're going to do these type of experiments. The first thing is you've got to plate the cells overnight. They have to be sticky. They have to stick to the plate. We're going to talk about plating in a minute. The other thing that you do, these cartridges arrive freeze-dried. You have to take the lid off, and you have to hydrate the cartridge. The little sensors on the end, they're in sort of a gel matrix, and the gel has to be hydrated the night before. So you put the cartridge in the calibration solution that comes with the kit. Then on the day of, you're going to take the cells and you're going to replace them with the media that you're going to do the experiment in. We'll talk more about media in a second. That's about an hour before the experiment. You load all of your various drugs, so the antimycin, the FCCP, the rotenone, the oligomycin. You put all those in the injection ports ready to go. You load the cartridge into the machine and you tell it, hey, calibrate the probes. And basically what it's doing there, it knows that the calibration solution at 37 degrees in room air at 600 feet above sea level in Rochester, New York, contains this much oxygen. And it does a single point calibration for the oxygen concentration. Then basically what happens is the green part, the cartridge, stays inside the machine. Your plate with your calibration solution gets kicked out, and the machine says, okay, now give me your cells. You then put the cells in, and the green measurement cartridge comes down on top of your cells. And that's when the experiment begins. The experiment itself takes about an hour and a half. So basically the workflow here is the night before you get this stuff ready, the day of, you're talking about an hour of messing around with multi-channel pipettas to get all the drugs set up, and then about an hour and a half to run the experiment. So plating, there are a couple of different types of plastic that are sold. They sell PS plates or PET. Different cells are going to stick differently. You can use a plate rotor to centrifuge them down. You can coat the plates. We use laminin. If you've got cells that don't really stick very well, you can use something called cell tack, these type of things to get the cells to stick. But the important thing is you've got to have a monolayer of cells at the bottom of the plate. The way you do that actually is you plate the cells in a small volume, allow them to stick, and then top up with the media. If you just plate the cells in the media, you're going to get cells all the way up the side of the plate here, and that's useless, right? The only ones that are going to get measured are the ones that are at the bottom. So it's a two-step plating process. You put them down, wait about 20 minutes, and then come back and top up with media. Cell number per well varies hugely, right? I mean, we use cardiomyocytes. We can get away with about 2,000 cells per well. If you're using platelets, you need probably 250,000 per well to get good rates. So this varies hugely depending on cell type, oxidative capacity. Cancer cells are going to do more glycolysis and less oxygen consumption. So your type of cell is going to be quite important to think about cell number. Media and substrates. One of the things we mentioned, right? This machine measures pH. There's no use having bicarbonate as a pH buffer, because then pH isn't going to change. How do you measure a change in pH if that pH change is being buffered by bicarbonate? Okay, so it's critical that the experiment itself takes place in a bicarbonate-free version of DMEM. So basically what you do is you buy the powder for this bicarb-free, glucose-free, substrate-free DMEM, and then you add back in all the various substrates that you need. And you can put in glutamine, pyruvate. We use fat. The heart is a fat-burning organ, so we use BSA conjugated palmitate. You can also use oleate or other different fatty acids. Glucose, lactate, ketone bodies, galactose. You can put in all these different substrates. And this is an example just from one of our previous papers where we were looking at the baseline rate and the FCCP stimulated rate. So we're looking at the baseline rate and the maximum rate. And in these particular cells, when we gave them glucose and glucose plus palmitate, they were capable of reaching, the knockout cells here were capable of reaching the same oxygen consumption as the wild type cells. But when we gave them palmitate alone, they were not capable of reaching that maximum rate. So that allowed us to say that these cells, this knockout cell, has a specific defect in fat oxidation. So those are the types of things you can do. And we didn't find differences when we provided any of these other things as the sole respiratory substrate. So you can play around with this type of stuff. It's kind of important to know, what are my cells burning as a fuel? And these are the type of things that you can do with that. In terms of the plate layout, there are a few other important things to know. It's 96 wells. That does not mean you can test 96 conditions. The biggest problem we see with this whole running this machine is everybody just comes along and says, oh, I can do a high throughput screen, 96 conditions per well. Actually, you've got to have at least 4 blanks. We prefer to have 6, typically on the corners. The reason for that is when the plungers come down, those wells are essentially shut off from temperature control. And temperature and oxygen are inversely related. That's why when you put a pot of water on the stove and turn up the heat, you get bubbles. That's oxygen and other gases coming out of solution as that water boils, right? That's why fish suffocate when the water is too hot in a river. And so what happens is as the plungers come down and the well gets sealed off, the wells are actually cooling down during the measurement. And so the solubility of oxygen is changing in real time. So you have to correct for that by having these empty wells that don't have any cells in them. And those wells are then used to correct for that change in oxygen that happens without cells there. You need at least 6 wells per condition, so how you arrange those on the plate is entirely up to you, whether you put them in groups, whether you put them randomized all over the plate. That's entirely up to you how you do that. And then there are a lot of other considerations. I would say if you're going to do one of these experiments, you've got to allow at least 2 or 3 plates to sort out the conditions. For example, FCCP, a lot of these drugs that we're adding are lipid soluble. And so the amount of lipid in the cell, if your cells have lipid droplets, that's going to soak up some of these things like FCCP, and you're going to need to add more of it in order to get uncoupling. But if you add too much, you can kill the cells. So a lot of people just want to jump in. You should allow at least 2 or 3 plates of data to play around with. Plating conditions. How many cells do I need? What's my dose response for FCCP? How much rotenone do I need in order to inhibit these cells but not kill them, right? There is a database on the Agilent website for different cell types, so you can go and find what are the conditions other people have used and papers that are published using all kind of different cell types. The drug stocks that you load up into the injection wells, they're typically about a 1 in 10 dilution in the same media as the cells. Seahorse will sell you a kit with oligomycin, all this other stuff. It's much easier to just buy the kit and make up those things rather than trying to buy the raw chemicals from Sigma and make up your own stocks. If you're going to be doing a lot of stuff, then yeah, do that. The cartridges cost about $55. They can't be reused. Lots of people say, oh my God, that's really expensive. Can I just wash it out afterwards? There's a couple of reasons not. One is it's plastic. All these drugs are lipid soluble. It's incredibly difficult to get the drugs off the cartridge. And secondly, the little hole down the middle that's right next to the injection ports, that's where the stainless steel rod goes that contains the fiber optics. If you don't get that cartridge completely dry and clean, you're putting those stainless steel rod into water and it's going to go rusty. And the person owning the machine is going to get very pissed off at you. So just do not attempt to reuse the cartridge, please. You can download the software to edit the methods offline and do everything and import it into Excel for data usage. The software is free to distribute. And be aware of edge effects. There's, these are 96 well plates. The wells on the edge of the plate behave differently than the wells in the middle of the plate due to temperature and diffusion gradients and all types of other things. So, be aware of that. one of the more important things that does need to be addressed are the raw data. You will see hundreds of papers in the literature where there's a seahorse trace and the y-axis says picomoles per mil. That is essentially meaningless. Doesn't mean anything. All it means is that well consumed that much oxygen. You have to normalize to cell number or protein or something, right? The key thing that we see a lot of people do is they come along, they plate the same number of cells the night before in every well, but then overnight, different cells and different wells are going to grow at different rates. And so in the morning, you've got a different number of cells in every well. There are a couple of ways you can do that. There is an algorithm in the Siligo where you can actually do cell counting on the plate after you've run the experiment. The other way, one of the reasons on the Seahorse here, we actually leave column 1 and column 12 blank because we want to use those for the standards for the protein assay later on. So you can do a protein assay on the plate. Also worth noting, one well is not a biological replicate. Lots of figures you will see in the literature where people just run something and they call it n equals 6, and in fact it turns out the entire figure in that paper came from one plate. That is not a biological replicate. That's maybe not even a technical replicate, right? So you're going to need to run lots of plates and average from day to day, and that makes normalization absolutely critical because every plate every day is going to have different growth rates, those types of things. And I think that's it. So we could save questions to the end, or do you want to go ahead and would that be easier, or? Okay, yeah, I can answer questions now if you like.
So can you have no content of serum in the medium?
Serum will affect metabolism. So some people do include serum. Typically, serum will have albumin in it, which will bind fatty acids, but it'll also bind a lot of the drugs, which are lipid soluble. So if you're going to have serum in the media, you're probably going to have to do dose responses to make sure that you've got enough FCCP, rotenone, antimycin, to overcome whatever is going to get bound by the serum. I would just say if you're going to have any serum there at all, do a dose response for all those drugs, right? If your cells are not happy without serum, then yeah, you're going to have to put it in. But generally, I mean, you will swap the cells into the media that the experiment is going to be done in. They'll be there for about an hour, and then the experiment takes about an hour and a half. So your cells need to be happy without serum for 2.5 hours. If they can last that long, then you're okay.
I think they were also saying that if you have a lot of serum, you can actually start foaming during the mix.
Yeah, if you have a lot of protein, yeah, then during the mixing, you'll get bubbles. Yeah.
I have a quick question.
Yeah.
Kind of practical. So for somebody who just wants to start using serum, because I think there are some differences between the setup. To me, all those Krebs cycles and everything makes sense of it. Overnight is impossible. So what would be your suggestions? What to pay attention? Let's say you run micro stresses, which parameters are probably would be the best to kind of follow up?
Yeah, I mean, the spare capacity is really the one that I think a lot of people concentrate on, right? I mean, so for example, the interpretation of that, like let's say you have a knockout or something, and the spare capacity is the same, but the basal respiration rate is different. So if the basal respiration is different, but the capacity is the same, that tells you those cells, for example, are maybe using less of their maximum capacity, and maybe they have less oxygen consumption contributing to ATP production. That will be an example of a cell that essentially has a decrease in work demand. Those are cells that are doing less work, and their ATP requirement has gone down. So that may not be, right? So when you've got this basal respiration rate, remember that is sort of an agglomerate of mitochondrial capacity, but also ATP demand. So if you see a change in the basal rate without a change in the maximum, that's not a mitochondrial change or problem, right? That's a change in energy demand of those cells, right?
Comparing two different kinds of cells, you want to get to similar basal respiration, or how?
It depends on, very often you'll see both the basal and the maximum will change. Sometimes you'll see, these 3 or 4 parameters can all, there are 35 different combinations of how they can show up. I mean, I think Roman's going to show a number of different outcomes. But yeah, beyond just really engaging with the data, I mean, if the book, this book is like the Bible in our lab. Yeah, Metabolism at a Glance. It's $40 on Amazon, and it's just this, every metabolic pathway you would ever want to use and see. We have like 3 copies of this in the lab. Super useful, sits on my desk all day long. Nobody can remember all of those pathways, right? I mean, you keep this on your desk and you just go and look at this book when you kind of, what was that metabolite? What pathways is that involved in again? Oh yeah, and yeah, this book is just the Bible for our lab.
We'll ask different people.
Yeah. All right.
Okay, any more questions? Thank you.
Thanks.
We can move on to the second talk. So we have Roman Elsif today to talk about his research using Seahorse, I guess. Right? Yep. So Roman got his MD in Moscow, National Medical University in Moscow, Russia. Moved to US in 2002, right? Straight to Rochester? Or
1996.
Oh, 1996.
96,
Oh,
yeah.
okay. It's a little bit of traveling.
Yeah.
And then faculties of U of R since 2006, and primary appointment in orthopedics, and secondary in pharmacology, physiology, and pathology. Also affiliated with Cancer Center, working on mitochondrial research, and the major focus role of bioenergetic mechanisms in cell fate decision, aging, and cancer. So a lot of different topics. Wrote in a lot of money, 6.5 million in grants, I guess. That tells something. So I'll just talk.
Thank you.
Sure.
Thank you, Wada. So yeah, thank you for inviting me. The series, I think it's very useful for people who just starting using these CART resources. In particular, Seahorse. I don't have too much to add in terms of theory or technical aspects of it, as Paul covered most of it. I guess what Paul haven't mentioned is not only did he have one of the first Seahorse machines, he was able to modify it. And actually, he was able to find a way to reuse the cartridges. And that probably prompted Seahorse and Agilent to change everything so that nobody does it anymore. And, they keep buying the stuff instead of modifying it. So anyways, so we're interested in how energy metabolism and cell fate connected, what's the crosstalk, what happens in aging with it. Our major focus is what's called bone marrow stromal cells, or aka mesenchymal stem cells, or bone marrow mesenchymal stem cells, aka skeletal stem cells. So none of these terms are correct. In fact, we know very little what the cells are and very hard to define them. There's a lot of confusion about it, but anyway, so those are the cells that you can isolate from the bone marrow, you can flush from the bone marrow, and they stick to plastic, and they're not hematopoietic, so they of mesenchymal lineage. And they're multipotent. So they, as you can see here, of course, in the bone marrow, in the majority of cells are hematopoietic cells, immune cells, but there's also stroma, and this stroma you can flush out, and you can grow in culture, or you can inject the cells into the animal, and you can get all these types of cells out of them. Osteoblasts, bone-forming cells that form bone, actually. You can get chondrocytes and a little bit of cartilage there, and also adipocytes, so a little bit of marrow, a little bit of fat, marrow fat. So here's again the diagram, simplified diagram, what the cells fates are, possible fates. Sometimes you hear other fates possible from them, but these are what we are interested in mostly. And in particular, osteoblasts and adipocytes. So adipogenic lineage versus osteogenic lineage. So as I said, the cells can make fat, bone marrow fat, or they can make osteoblasts, which make bone. So what happens in aging, and this is the example of what happens in mice, right? When you look at the micro CT or computer tomography scan of mouse bones, and this is the sagittal section through the tibia and femur. Inside you see this cavity, which is the bone marrow, where the bone marrow is. And if you compare 3 and 18 months, you can see how bone tissue goes down, it decreases. So there's a loss of bone, or osteoporosis. The same thing happens in humans. Cortical bone becomes thinner. The same thing happens in femur. But then if you stain the samples with osmium tetroxide, it labels fat, basically. And you can see, so this time you do micro CT4, and you can highlight the fat tissue. You compare young and aged mouse, the aged one is full of fat. So it looks like, and this is the function of BMSCs, right? So this bone marrow stromal cells, either they become osteoblasts and make bone, or they become bone marrow fat cells. And so there's an obvious fate switch in aging. So BMSCs, instead of forming bone through osteogenic lineage, they form fat. And this is one of the features of aging, and this is one of the pathogenic mechanisms of bone loss in aging. So our first question when we started studying it is, what are the metabolic preferences of BMSCs and their progeny, osteoblasts versus bone marrow adipocytes? And what we know about stem cells in general is, or various kinds of stem cells, is that when they are undifferentiated, they're mostly glycolytic. Well, first of all, they will less metabolically active, but they make the majority of the ATP through glycolysis. But then when they differentiate, most of the cell types, they switch to oxphos. And then if you de-differentiate the cells or make induced pluripotent stem cells, they switch back to glycolysis. So there's a tight link between state of differentiation or stemness and oxphos and glycolysis, so energy metabolism in general. So what we mostly do in the lab, we study just various lineages, and we differentiate the cells in vitro. So first of all, we plate the cells, we grow them, and then there's 2 ways to do it. Then we measure their metabolism using Seahorse. But there's 2 ways to do it. One is you differentiate on the plate for a certain number of days, and then a day before you reseed on the Seahorse plate. And this is, sometimes you have to do it this way, because if cells grow too fast, you cannot just seed them. And the second way is actually you just grow them, and then you seed them on the Seahorse plate, and you differentiate on the plate. But if the cell growth is an issue, you have to do it the very first way. Okay, so again, it depends on your cell type. You have to play it by ear. And usually what we do is, some wells are, undifferentiated cells, and then we see the other wells where the cells are differentiated. And then we're just doing this BOFA algorithm using Seahorse. Some technical points, although Paul covered most of them, but I guess one thing is that DMAM is without phenol red. We found that the phenol red can interfere with this fluorescent probes. Again, it's Paul mentioned that DMAM powder, and that's the best way to do it, right? So you can get the powder, you can weigh out everything, and you can manipulate the media as the way you want it. It's actually not a big deal. I mean, if you cannot mix a media, then I don't know if you need to continue with science, right? So we all need, have to know how to do it. Okay, so we just mix the media and add the fuels, of course. One thing to pay attention to is glucose. In most media compositions, glucose is very high, right? So it's like 25 millimolar, which is super high. If you're interested in what happens in vivo, or if you want to mimic the in vivo situation, you have to use physiological amount of glucose and other fuels, which is around 5, some little less, little more, 5 millimolar. And again, it's very easy to make. If you're using 25, you can initiate what's called the Crabtree effect, which is inhibition of OXPHOS by high glucose. A lot of media compositions and kits that Agilent sells, they have 25 millimolar glucose, so be careful with that. And again, physiological glutamine, no pyruvate, because pyruvate in vivo, in plasma concentration of pyruvate is actually very low. I mean, you can add a little bit if you're interested in it, but we don't use any pyruvate because we don't want to mess around with the metabolism, right? So if you have pyruvate, why do you need to use anything else, basically? And then we do add a little bit of FBS sometimes don't, but it does help. It adds a little bit of the growth factors and a little bit of fatty acids that are required in most cases. And then we get the curve like this, right? So it's if you look at it, this is how undifferentiated cells behave. So we do this regular, BOFA that Paul mentioned. We use oligomycin, FCPA, antimycin A, rotenone, I forgot to mention here. Basal respiration, then ATP linked, and you derive all these parameters from it. But this is how the curves look. So this is undifferentiated in blue, and then osteogenically induced in red. So you can see how the basal respiration even significantly higher compared to undifferentiated cells. And then maximal respiration also goes up. ATP linked respiration here, the difference between this rate and this rate here is much higher than between this and this. And this is what happens with ECAR, which is a glycolytic rate. So it doesn't change much if you differentiate the cells into osteogenic lineage. So another example I wanted to give you is we're interested in how is metabolism activated during BMSC differentiation into osteogenic lineage. And then we had to remember how activation of the cells happens. And this happens because of bone remodeling, which happens constantly, right? So there's a constant remodeling of the bone within the bone marrow. Osteoclasts, which are bone resorbing cells, they just resorb bone constantly, in some places more actively, in other places less actively. But when they do it, they basically resorb like microfractures or some older bone regions. And when they do it, they release a bunch of growth factors and hormones that are stored in the bone tissue and bone matrix by osteoblasts, such as TGF-beta, IGF-1, and then BMP and Wnt also getting released. And those are all the factors were shown to affect BMSCs, activate them, and force them to differentiate. So that's why we decided to try what is the effect of this BMP, Wnt, TGF-beta on cell metabolism, or BMSC metabolism. We also use this osteogenic media, which is just ascorbate and inorganic phosphate, beta-glycerol phosphate. And this is the very easy way, cheap way to force cells towards differentiation, because when cells have this, they start pumping out their own BMP and Wnt and kind of activate each other and themselves. So this is what we tested, and it was very actually convenient to use this type of graph, right? So when you measure ECAR and OCAR, then you can use the so-called energy vector plot, right? So what happens is that you plot OCAR versus ECAR, and then you plot it baseline rate versus activated rate of OCAR and ECAR, and this results in this vector. And what you can, and looking at this vector, where it goes, basically, you can determine whether there's aerobic or glycolytic, cell becomes more aerobic or glycolytic. Because if it goes here, it's just equally OCAR and ECAR activation is equal, so it's like just more energetic cell. If it goes this way, it's more aerobic. If it goes this way, it's more glycolytic. And so what we found basically is that BMP and Wnt and of course osteogenic media are the ones that are most active in activating OXPHOS in the cells. So again, there's a couple of ways to do it. So you can take the plate and you can pre-treat cells first and then measure, or you can add it if possible. If you want to look at the immediate effect, immediate response, you can add in one of the ports, you can add your drug of interest or hormone or cytokine, and then it'll inject, and you can almost in real time you can see the response, right? And so we did it both ways basically. What we found is that all of this osteogenic media, BMP2 or Wnt3A, if you add it, you force cells to go more towards aerobic, oxidative metabolism. So basically, you can see how the vector goes more like here instead of more towards more glycolytic. And this is uninduced cell. This is the induced cells. And this is pretty much the same response we got with all three. We also figured out that this is all goes through AKT, one of the major signaling mechanisms. So if you block AKT, then the cells don't respond and they just behave the same as uninduced cells, basically. You can see here on the plot. So this is, again, very convenient way of studying cell metabolism and what regulates it. If we force the cells to differentiate into adipogenic lineage, right? So we can, again, do it on the plate. This is what happens. So this is undifferentiated cell here, undifferentiated cells, and these are adipogenically induced. So the basal respiration is up, maximum respiration is up, Actually, here it's also a little higher, non-mitochondrial respiration. But in contrast to osteogenic lineage, you also see a major activation of glycolysis. Okay, so you can see here in red how glycolysis is much higher. So then I'll give you a typical example of what parameters we derive from it. So Paul again mentioned some that from responses to these drugs, you can figure out basal respiration, ATP-linked respiration, maximal respiration. And that's what we've done here. So all of these parameters you can get just from, one run. But of course, you have to do more than one run to get biological replicates. And this is what happens. So we see upregulation of OCAR, oxidative phosphorylation, but even more so of glycolysis in adipogenically induced cells. And one thing is, so there are some, some of the parameters are still kind of maybe a little controversial. So some people say you can derive certain parameter from it. Some people say you don't. But there are papers that can, there are protocols that suggest how you can, for example, calculate mitochondrial ATP versus glycolytic ATP and figure out contribution of both towards metabolism. So I would recommend everyone to look for papers by Martin Brand, Dave Nichols. I don't know, for some reason all these guys are British. Right, Paul? And they figured out the way to do it and to actually take into account all this, acidification due to basically non-glycolytic pathway due to TCA cycle and all that. And then there's an algorithm and formula to basically figure out mitochondrial ATP versus glycolytic ATP. And here's what happens. So we see there's much more ATP produced in adipogenically induced cells, but most of it is actually due to glycolysis. And it becomes even more glycolytic here at day 7 of adipogenic differentiation. So the OXPHOS contribution shrinks and glycolytic contribution expands. So that's the example I wanted to show you. So a lot of different graphs you can put. So this is really well-developed, I would say, technology and then analysis of it. One other example I wanted to show you, although Paul showed some of it, is we wanted to figure out what's the most important fuel for these cells. So again, you have to use physiological concentrations of those, and of course you have to make it yourself, mix it yourself in most cases. And so we looked at individual fuels such as glucose only, glutamine only, palmitate, fatty acid only, of course with BSA, with albumin complex, and then we used combinations of these different fuels. And so here is the basal respiration, here's maximal respiration, and the basal respiration is where we saw most of the changes, right? So the palmitate here, it looks like it's the most efficient fuel for BMSCs or for mesenchymal lineage cells. And then it has been shown by other groups that this is very prominent way. So these cells these cells are most efficient when they have palmitate. But it looks like, in going to other assays that we've done, glutamine is probably the main fuel for these cells, not fatty acids, not glucose. And one evidence here is that we saw is that when you have glutamine in complex with palmitate, they don't respire that much. So it looks like maybe outcompetes palmitate for some reason, and basically it most likely that's what drives respiration in the cells. So again, so this is very convenient way to figure out all these parameters. Few suggestions in addition to Paul's is again, for mimicking in vivo conditions, use physiological concentrations of glucose and other fuels. In contrast to most in vitro media, in vivo concentration of pyruvate is very low, so have it in mind when you design your experiment. You need to do pilot experiments to determine the optimal cell number and also optimal concentrations of FCCP and other drugs. And in addition to normalizing to protein or to cell number, it's actually recommended to also find out if the amount of mitochondria change in your cells, right? So if you use different conditions, or if you differentiate your cells and look at differentiated versus undifferentiated, OCAR may be affected. The cell number may be still the same, but the number of mitochondria may be different, and mitochondrial mass may be different. And it's quite actually easy to measure that. You can measure mitochondrial DNA and use it as a normalizer, or you can measure mitochondrial mass using fluorescent probes and take it into account. Okay, so here's my lab who did most of the work, and thank you.
I have one question.
Yeah.
So can you actually use this technology to sort of figure out the stemness of the cells or differentiating stage? So how sensitive it is. So I'm thinking, you have to grow those cells. So if you have number of passages, can you actually catch using this when they become too differentiated?
Well, yeah, actually, if you know for sure that this is oxphos or glycolysis is activated during differentiation, and you see the changes between different passages, that may be an indicator that they start spontaneously differentiate. Other thing that can happen is they can become senescent, right? So, and then of course senescence also associated with just inhibition of metabolism. So you will catch it using Seahorse, but I mean, of course, you need to measure other things, right? Not just metabolism.
Well, I'm just thinking because it's used a lot to check like CAR T cells, for example, how well they actually going to perform. Are they really effector cells or more like memory cells, and that's actually useful for therapies. Yeah. Okay, no more questions. Okay, so we are kind of late, so I will. Thank you. The speakers again. I have a few things to say. I won't go through my presentation anymore, but we actually are starting a service, full service for the Seahorse. If you are afraid to try it, or you don't want to invest all the time in buying reagents and everything, come talk to us and we can figure out a way so we can actually, you provide the cells and we're gonna do the okay? Well, we calculated it might be, if you're not planning to do a lot,